Assalamu alaikum my dear students. Hope you are fine by the grace of Almighty Allah. Welcome to NIV's Home School. I am Nasima Akter, Assistant Teacher of Science, National Ideal English Version School, Morning Shift. Dear students, today I am going to take a biology class for class 10 students. Dear students, our today's topic is Chapter 11, Part 2. Let's see today's topic from here. Our today's topic is Chapter 11, Part 2. And here is lecture number 2. SW number is also 2. So dear students, let's begin the class. In our previous class we have already read some part of this chapter. Today we will start from this page 233 page. And here is pollination. Pollination is also called the linking of pollen. Pollination is a precondition for the development of fruits and seeds. The transfer of pollen from the anther to the stigma of the same flower or to the stigma of another plant of the same species is pollination. So this is the definition of pollination. Pollination is two types, self-pollination and cross-pollination. Self-pollination. On the same flower or on two flowers of the same plant, transfer of pollen can occur. This is called self-pollination. For example, brassica, cucumber and datura usually pollinate through this way. Wastage of pollen is less. The process is not depend on a carrier and the occurrence of pollination is ensured. The plant that develops in this way cannot make change to their characters, and so the features of the species can be maintained. This way a species can maintain purity. No new characters appear in the new generation of plants. The newly born plants breed seeds with less vigor. The ability of adaptation in the new plant is reduced, which means this species can be extinct. Now cross-pollination, when the attachment of pollen occurs in between two distinct flowers of the same species, it is called cross-pollination. Such as cotton tree, papayas pollinate this way. And here is the picture of self-pollination. When a pollination occurs in the same plant, this is called self-pollination, and here is cross-pollination. When pollination occurs between two plants, this is called cross-pollination, and they must be same species. Now come to here this page. This is 234 page. New characters emerge through cross-pollination. The rate of germination goes up and much more vigorous seeds are produced through this way of pollination. As the pollination occurs in between two different plants, the seed produced in the way is born with new characteristics. The plant which grows from the seed also has new characteristics. This is why new varieties of those plants emerge. As it is a carrier-dependent process, pollination is not ensured, and large waste of pollen is a consequence of these types of pollination. So the purity of the species is impaired. Now medium of pollination. In most cases, a medium of accomplishes the transfer of pollen. The carrier which carries pollen is called the pollen carrier. Air, water, insects and flies, birds, vampires, snails and even humans may be the media of pollination. To ensure nectar from a flower or after attraction to the charming color of the flower, the carriers travel from one flower to another. When the carrier sits on the flower, the pollen attaches to it. To obtain the aid of the carrier, some plant adaptations are remarkable. Pollinated. Insect pollinated flowers are big with colored nectar secreting glands. In these flowers, the fragrance of wet fluid is alluring. For example, hibiscus, gourd, mustard, etc. Air pollinated flowers are lightweight and do not have nectar secreting glands. The flower has no fragrance, can easily wander in the air. Their stigmas are branched and sticky and sometimes feathery. Branched stigmas will attain pollen from the air. For example, Oryza sativa. Now come to here, this page. Water pollinated plants are small and lightweight, so they can float in the water. They have no fragrance. The petiole in the female flower is longer than that of the male. Mature male flowers detach from the petioles, float in the water, and after coming in contact with the female flowers, they accomplish pollination. For example, Vallisneria. Animal pollinated flowers are usually large in size. If they are small, they are arranged in the inflorescence. They have attractive colors. Fragrance may be present or absent in them, such as cotton tree and taro. Now here is microsporogenesis. Development of microsporogenesis. A pollen is the first cell of gametophyte. Pollen is the first cell of gametophyte. Pollen mother cell is twice diploid. Produce 4 pollen haploid by meiosis cell division. Soon after becoming mature, harboring in the pollen sac starts to germinate. The nucleus of the pollen gets divided through mitotic division and 2 cells, one large and another small, are formed. The large one is called the tube cell and the small one is called the generative cell. The tube cell turns into the pollen tube and 2 male gametes are produced. Division of the generative cell takes place in the pollen or pollen tube. Students, here is the picture of pollen and here is the extine and intine layer, and this is the generative nucleus, and here is it grows, here is tube nucleus, and here is pollen tube, and this one is 2 male gamete. Now megasporogenesis. Near the micropyle being nourished in the nucleus of an ovule, a cell starts to become larger. Its protoplasm is dense and the nucleus is comparatively large. The cell divides into 4 haploid cells through meiotic division. Every cell except the smallest one gets disintegrated, growing gradually. The larger cell matured into a embryo sac. The nucleus of the cell is haploid. The nucleus of the cell is haploid. The nuclei are evolved as the nucleus divides. These 2 nuclei take position in 2 opposite poles. Next, these 2 nuclei consecutively divide twice and result in 4 nuclei. And here is the phases of this division. You can see in the last stage, here is total, this one is the female gametophyte development, and here is 3 nucleus, this is called antipodal cell, and here is 2 nucleus, this is secondary nucleus, and here is another pole, here is also 3 nucleus, and the middle of this nucleus, this is called egg, and the side here is 2 nucleus, this is called synergic cell. Now come to here, in the next stage 2 nuclei from the 2 poles come to the middle of the cell and get fused to cause the emergence of 2 haploid secondary nuclei. The nuclei in the 2 poles turn into cells with some amount of cytoplasm. Collectively, the combined structure of the three cells near the micropyle is called the egg apparatus. In between the cell, the one occupying the middle place is the ovum, which is a little larger. This is called an egg cell and the other two are called synergy cell. The cell in the opposite pole to the egg apparatus are called antipodal cell. This is how the process of developing the embryo sac is done. Now, fertilization. Through pollination, the mature pollen lands on stigma of a carpel. Then the pollen tube grows, penetrating the style and absorbs some liquid. Once the tips of the extended pollen tube enter the embryo, it ejects two male gametes into the embryo sac. One of them meeting with the egg causes the emergence of a zygote. The other male gamete mixing with the secondary nuclei develop triploid endosperm cell. Now come to here. This is the figure of life cycle of a flowering plant, and this is a plant, and this is sporophyte, that means twice nucleus, and from this plant here is flower, and we can see here is part of this flower, anther, petal, filament, ovary, ovule, and from flower here is female gametophyte will be produced from here. This is megasporangium, and after meiosis here will be megaspore, and then mitosis. Again it will divided by mitosis, cell division, and here is embryo sac. This is embryo sac, and from this anther here will be microsporangium, and next here is pollen mother cell. Pollen mother cell will be twice-n, that means diploid, and after meiosis it will be 4 daughter pollen cell or microspore haploid. Now here is the daughter pollen cell or pollen grain. N number nucleus, and this is haploid also. And then next step here is embryo sac, pollen tube, and after fertilization here is embryo, seed coat, endosperm. Again from seed there will be new plant grow. So this is the life cycle of flowering plant. The two fusions, mixing of a male gamete with an egg and mixing of another male gamete with secondary nuclei take place at about the same time. This phenomenon is called double fertilization. Now development of new sporophyte. A zygote is the first cell of sporophyte. Two cells are produced after its first division. At the same time, the flourishing of the endosperm is also initiated. A zygote divides transversely. The cell towards the micropyle is called the basal cell, and the cell towards the center of the embryo sac is called the apical cell. The division of these two cells continues simultaneously. Gradually, apical cell turns into the embryo. The suspensor also gets developed. Consequently, the cotyledon, radicle, and plumule are developed one after another, and the secondary nuclei start to make the endosperm. The cell in a endosperm are triploid. The cell in an endosperm are triploid, meaning they possess 3 sets of chromosomes. This is very important for your MCQ. At maturity, the ovule with the endosperm and the embryo turns into a seed. As the seed germinates, a complete sporophyte is developed. Students, now look at here. This is the figure of structure of ovule and fertilization, and here is you can see this is nucleus tissue, antipodal cell, secondary nucleus, embryo sac, this is, and this one is egg, and here is synergid egg, two synergid egg, and here is inner membrane, and here is outer membrane. Here is inner membrane, this one is the inner membrane, and this one is the outer membrane, and here is the pore, this one is the micropyle, and here is, this one is called helium, and funiculus, this one, and here is the male gamete, and this one is ovary, ovule, and pollen tube, this is pollen tube, stigma, and pollen. The sporophyte and gametophyte run in a repeating way one after another through the life cycle of a flowering plant. Origin of fruits. When we think about fruits, the image of mango, jackfruit, lychee, banana, grape, apple, guava, sabeda, etc. appear in our mind. Bottle gourd, jhinga, pointed gourd are also fruit, though they are called vegetables. In fact, they are all fruits. The process of the formation of fruits began immediately after the completing of fertilization. Fertilization initiates stimulation in the ovary to develop fruits steadily, and ultimately ovules are turned into seeds. The ovary after fertilization turns into the fruit. If only the ovary turns into a fruit, it is called a true fruit. Mango and berries are example of true fruits. When along with the ovary, other parts of a flower get matured, these types of fruits is called false fruit. For example, apple, chalta are examples of false fruit. All the true and false fruits are grouped into three categories: fleshy fruits, aggregated fruits, and the compound fruits. Students, I have finished my today's topic. Now I will give you HW. Our today's HW number is 2 number HW, and your HW will be make 5 MCQ. From this part, make 5 MCQ from today's topic. So dear students, I hope you have understood today's topic. Keep practice it at home and do your assigned SW in your SW copy and submit your SW on every Saturday. So dear students, stay home and we'll see you again in our